Thank you. Hello everyone, my name is Ziaeddin Alborzi. I'm a PhD student at the University of Lorraine in Nancy, and today I'm going to talk about a project that I did as an internship in ABI with the name of Automatic Generation of Functional Annotation Rule using Inferred Go Domain Associations. Our project has two phases. The first phase is associating Go terms to domain directly and then use them for propagating function to all sequences that they have. They don't have any function or they lack annotation. Here is the outline of my talk. First I introduce, then I talk about the Go domain miner for assigning Go terms to domain directly, and then I use those Go domain associations. And finally, I participated in KAFA challenge. I show you my results, and finally acknowledgement. As you all know, protein are the yes. Oh, how can I go? I don't know. I have no idea.
It's advancing up here. Let's
Is this a web presentation?
No, it's slides.
actually he's not in slide mode, so that might, is that a different set of, is that like off a server or something? No.
Actually, I was telling that proteins are the macromolecule in the body that they do the function, and this function can be actually represented in different ontology like Go terms, and usually these functions are done inside the conserve area, inside the sequence. They call it domain, and they have different classification like PFAM said. Here I have an image. I show you that usually, Go terms are associated with whole sequence and they are not associated with the domains. What we are going to do is assigning the Go terms in different situations directly to domains, and then use these Go domain associations for functional annotation of whole sequence. The Go domain miner actually has been published, and the similar method for EC has been published here. You can see that. We say for any protein database that we can create this tripartite graph, which Go terms and domains are associated indirectly through proteins. And what we are interested coming up with this direct association between Go terms and domains, which can be multiple to multiple. And to do that, first we need to divide this tripartite graph into two-partite graph. And then each bipartite graph can be represented as a bijacency matrix, incident matrix, and this matrix represents Go term and sequences. It's a matrix of Go and sequence matrix. If there is a link between Go and sequence, we put 1, otherwise it's 0. And since we are in the space, we can find this cosine similarity between each Go term and domain using the sequences. And finally, we come up with a Go domain matrix, which each cell here shows the similarity score between the Go and the domain. Usually we have several data sets, input database, or they can be divided into several sections based on their features. For example, in our problem, we have SIFT, which has been divided into two, SIFT IEI and SIFT experimental, and Swiss Prot and both the same. And we introduced a consensus score, which is a weighted average for each of these similarity score. And the formula is here, it's a weighted average. And to calculate that, we use a rock plot, and we varied each weights from 1 to 10 in the steps of 1, and in each step we calculated the rock plot and we calculated the AUC, and at the end of the process the best AUC gives us the best combination of the values. Now that we have the consensus score for all the possible Go term to domains, the associations, we know most of them are false positive. In order to remove them, we rank them, and we rank based on the consensus score, and we create a negative data set by shuffling from the tripartite graph and creating Go domain which are wrong. And then we vary the consensus score from 1 to 0.01 in the steps of 0.001, and for each step we calculate the F measure for the training data set, and we find the best F measure. And the best F measure gives us the best threshold. So we usually get around 5% of top data. Now that we have the Go domain association, we can, they are ready to be used for Go sequence prediction. And we simply can say if a sequence has a domain, we can annotate the sequence with associated Go terms. But there are three issues. The first of all is we did not take into account the taxonomic information. Second of all, one function can be done by several domains. And the last one is different domain classification. Some classification like PFAM are all-inclusive. We can find it in almost all the sequences, but some of them are very good, like TigerFam and SED for just one-to-one Go domain association. So the solution would be a systematic way to handle these three issues. We devise this a method, we call it autoproof annotator. In the first step, we find the Go domain associations using the Go domain miner to assign Go to all these 9 domains, PFAM, TigerFam, CAT, SED, Panther, Smart, all of them. And in the second step, we try to define a rule. The rule is something that once the left-hand side is met, the right-hand side will be initiated. So there are 2 examples here. We say if the sequence belongs to bacteria and there's a TigerFam, this one, then we can assign this Go term for molecular function or for BIP, for biological process. In a very simple algorithm, here I explain how we created this rule. We get all the domains which are associated to the Go terms, and out of those, we create all the subsets, possible subsets. We have a limit of 5, and then after we add the taxon that we want to be annotated. We get all the list of taxon from SwissProt and add it to this Go term, to this left-hand side, and we say the right-hand side should be Go term. Since we are in the domain of rule mining, obviously, we can define 2 metrics which can be used for further filtering and finding the quality of the rules. If we assume that the database is SwissProt and transaction is a combination of domains, taxon, and Go terms, for each candidate rule, we calculated the support, which is simply frequency of happening that rule inside the SwissProt, and confidence is to show how confident that rule is and how trustworthy is that. We use these 2 metrics for further filtering. Here, we have 2 levels of filtering. The first one is high support. It can be 5, 50, 100. Of course, with a higher support, you can get the better rules, which are more trustworthy, and the high confidence. The high confidence of 1 I usually use because I do not want to predict something more than what we have in SwissProt. I trust on SwissProt for now. Okay, here is some statistic on our rules, the combination of the rules. You see if we keep the confidence 1 and confidence 0.5, there is not much differences between these number of the combination between rules. The distinct taxon is also quite similar, and the prediction rule is the number of the Go term that we can assign actually on the right-hand side. The only thing which is very different here, and it's hidden inside that, is a combination of all these 3 together. So of course, for confidence 1, we have less rules, and for confidence more than 0.05, we have much more. Here I did 1 cross-validation, a very small cross-validation, 5-fold cross-validation inside the SwissProt, and the average recall was 56. I can guess because some sequence, they have some domain that actually, if they happen in the test, but we don't have it in the training, we cannot get it. But precision is not bad, 98. And then I compared it with the result inside the Tremble. I got all the rules with the confidence of 1. For example, here for virus, you see if we increase the, if we decrease the confidence from 1 to 0, we do not get much coverage than what we have in confidence 1. So we trust on our confidence 1 score. Here one example. I say in the rule 1, if you have, if a sequence have this PFAM, this cat superfamily, and it's from Mammalia, we can assign this Go term. Inside the SwissProt, there are 5 hits. There are 5 sequences that all has been annotated by this Go term, and inside the Tremble, there are 47 sequences, which just one of them has been annotated by this Go term, and 7 of them has been annotated with the ancestor of this Go term, and 39 of them can be annotated. And the second rule is very simple. CDD, as I said, is very good for direct association between domain and Go term and even EC. And we say if the sequence has this domain and it's from protobacteria, we can assign this Go term. As you see, there are 103 sequences hits inside the Swiss prot. The data back to 2-3 months ago, if it should be very similar still. And the Tremble annotation, there are 1,930 sequences, which 190 of them are not annotated with this Go term, and for 570 of them, roughly we can find more specific Go term. I took part in the KAFA challenge. You all know what is KAFA challenge. It's a critical assessment of the functional annotation algorithm. They provide a huge number of, not huge, 130,000 K sequences, and the output would be annotation, the Go sequences at the end. The number of the sequences was 130,000 in total. We ran the interpreter scan on them, and 121 of them had a domain that we needed. And the number of prediction you see here, for around 100,000 of them, we could find molecular function, biological function, BP and CC, with quite diverse number of Go terms. And based on, since we are using the domain-based method, we can find usually on average 2 or 3 Go terms for each sequence. Here is one example. I randomly chose that, but it was very good. This is a target. This is a target. I think so. Actually, this protein is a target in KAFA, and I found it in the Swiss prot. In the Swiss prot, there are 3 Go terms, and based on our rules, we found exact match. And also, there is no CC in the Swiss prot, but we found with a confidence of 0.91 CC. At the end, I'm going to thank my supervisor, Dave Ritchie and Mary Dominic, and my colleague Saber, and Maria for accepting me for this internship, and my friends Rabi and Alex for their support and their amazing ideas. And also, I'm going to thank NSF for partially providing the travel fellowship. Thank you. If you have any question which takes a lot of time, I will be at this poster. Please come.
We have time for questions.
Okay, thank you for the talk. How many terms in total can you predict with your rules?
I cannot hear you.
How many terms in total you can predict with your rules?
For the KAFA or for the term?
Like
Overall?
Overall, yes.
Or it should be here. You can just sum all these. It's around 16,000.
16,000.
Yes.
Thank you.
Question.
I'd love to see the number of the
Sorry? Okay.
Yes.
Have you considered any other input data than domains, like disordered regions, binding sites, or
I'm going to do that for the future work, but this is the main project. I try to actually stick with just Go and domain.
Yeah.
In the future, probably I do that.
Yeah, okay,
If I have
good.
enough funding or good postdoc.
So
Yes.
I assumed you looked at the distribution of the number of sequences you annotated with each term.
Where? This one?
No, this is a precision recall.
Yes.
So the actual sequences, the distribution of sequences, but what do you mean with distribution of sequences?
Like, so the number of terms, it'd be useful to see the number of times you annotated a sequence. The number of times you annotated a specific sequence to a term. So some
for
terms
KAFA,
are
you
going
mean?
to have, yeah.
Oh, for KAFA it's at the end here.
Yeah, Close enough. We can talk about it offline. It's not a big deal.
Okay.
Are there other questions? Great. Let's thank our speaker again.
Thank you.